How can protein formulations have a high concentration and at the same time a volume low enough for subcutaneous injections? In the past this was difficult and sometimes even impossible. We at Merck have created a solution for that. I'm Tobias Rosenkranz and I would like to introduce to you today our solution, the Viscosity Reduction Platform. Products that are available for intravenous administration are typically low concentration solutions. They are administered to patients, usually in large volumes, for example, through an infusion bag. That means the patient has to stay a certain period of time in a hospital chair, which is a rather inconvenient solution. A subcutaneous administration would be much more convenient for the patient, but it means a much smaller volume is required and the protein concentration has to be increased. This, however, comes with a significantly increased solution viscosity, which is the main limiting factor in creating a subcutaneously administrable protein formulation. If the solution viscosity becomes too high, it becomes too difficult to extract the solution from the syringe and to inject the drug into a patient. Now let's take a closer look at when and why proteins become viscous and take a look at the case studies that we've conducted in my lab. We looked at a number of different antibodies and when and how they become viscous. On the x-axis you see the protein concentration, on the y-axis you see the viscosity, and typically a solution is no longer administrable when the viscosity increases above 20 or 25 millipascal seconds. And you can see that some proteins already become too viscous at a concentration between 100 and 200 milligrams per milliliter. These proteins have a certain tendency to self-interact and form transient clusters that ultimately give rise to protein viscosity. Other proteins that become viscous only above 200 mg per ml also form these transient clusters, but here the next neighbor distance in between the molecules is simply too short and too narrow, similar like visiting a crowded football stadium when you are bound to interact with your direct neighbors. Here these transient clusters are formed by crowding, by the high concentration, and ultimately also give rise to protein viscosity. So how can we overcome this issue? Let me tell you how we went about in my lab to find a solution. First of all, we looked at the effect of different excipients and excipient combinations on proteins, on antibodies. This is important because the factors and the forces that cause protein viscosity on the one hand are the same that cause the protein to be stable as well. And this has led us to an important conclusion. We have to combine excipients in a way that protein viscosity is reduced, but that we can also stabilize the protein and balance these two factors, protein viscosity versus protein stability. We typically combine two different functional groups of excipients, amino acids on the one hand and anionic excipients on the other hand, a total of 9 functional combinations. When combining an amino acid which typically not only reduces viscosity but also has a stabilizing effect on proteins with an anionic excipient which has a much stronger viscosity reducing effect but also a higher risk of destabilizing the antibody, we create a synergy in a sense that a lower viscosity can be reached because both excipients work better together than alone, and at the same time stability of the antibody can be maintained. Let me show you the potential of these excipient combinations using the blockbuster antibody infliximab as an example. If we increase the concentration of infliximab to a concentration that is relevant for subcutaneous administration, we see that the viscosity increases beyond the level of injectability. If we then add arginine, a typically used industry standard, to the formulation, the viscosity doesn't change very much. And when we then apply the viscosity reduction platform, we see that we can create a total of 5 different combinations that would be injectable. And infliximab is only one example. As every protein is different and has different characteristics, it's important to have a portfolio to choose from. As you may have guessed, reducing protein viscosity is already beneficial during the manufacturing process, for example during tangential flow filtration, TFF, where the protein concentration is increased to its final value. During this step, elevated protein viscosities can lead to an elevated system pressure, which ultimately can make it impossible to reach the desired target concentration. Adding the excipient combinations does not only enable reaching the desired target concentration, it can also make the entire manufacturing process more economically attractive. Summing up, I would like to re-emphasize the heart piece of our technology: superior viscosity reduction by using excipient combinations and at the same time maintaining protein stability. And this can be brought together by enabling you to create a subcutaneous protein formulation. And this all led to the development of the Viscosity Reduction Platform. Are you curious for more details about our solution? Then I would like to invite you to take a closer look at the Viscosity Reduction Platform and explore more about highly concentrated protein formulations and on how to create formulations from intravenous to a subcutaneous route. Looking forward to talk to you soon.